everyone. Hope you're all having a really good day. Myself Tanuja, and I'll be discussing biology with you, right? So today I'll be discussing the first chapter of +2 biology, which is sexual reproduction in flowering plants. So this is your first chapter in your +2 syllabus, but actually in the syllabus you had before, you had one more chapter before this chapter. But right now that chapter is not there. That chapter is deleted from your syllabus, and you'll be having sexual reproduction in flowering plants as your first chapter. This is a very easy chapter compared to other chapters, but this is kind of lengthy. Okay, it's kind of a big chapter, so you'll have different parts coming along. But the only thing that you have to do is stick with me, write notes. Okay, write clear notes on the chapter, like notes in any language that you can understand. Start writing notes, lecture notes or whatever, using a guide or a textbook or even the class that I'm taking right now. Write proper notes and just hear the story with me. Okay, it's basically a story. It's basically a story of how a plant reproduces. Okay, it's just a story, okay, of how a plant reproduces. With the help of what does a plant reproduce? Okay, so here in this chapter we'll be discussing about the reproduction in plants. And when I talk about plants, we are not talking about all plants in general. Okay, we are talking about flowering plants, the plants that are able to flower. Why does sexual reproduction only happen in flowering plants? Is it so? I mean, is sexual reproduction only happening in flowering plants? Yes, sexual reproduction only takes place in flowering plants. Why do you think it is? Why do you think that sexual reproduction in particular only takes place in flowering plants? It's because flower is the sexual reproduction of a plant. So when we look at a plant, there are different organs, right? There are leaves, stems, roots, and there are other organs as well, right? But flower is another organ of a plant which helps it in reproduction. Okay, a flower helps the plant in reproduction, in sexual reproduction to be precise. Plants have other modes of reproduction as well. Plants have a sexual reproduction, plants have vegetative reproduction. But in the case of sexual reproduction, so what do you mean by sexual reproduction? Let us revise, okay? You may already know this from your +1 chapters, but still, let us revise, okay? Sexual reproduction technically means reproduction with the involvement of two parents, right? So there will be a male parent and there will be a female parent. So both these parents together get involved in sexual reproduction and produce a new offspring. So that's what sexual reproduction is, right? Also, if you remember, sexual reproduction always happens in the presence of gametes, right? You'll have a male gamete and you'll have a female gamete. Both these gametes will fuse or fertilize to form a zygote. That's what usually takes place in any typical sexual reproduction process. Okay, cool. So now you know what sexual reproduction is, and now you know what flowering plants is, right? Cool. In this chapter, that's what we'll be discussing. We'll be discussing, at the beginning, we'll be discussing the morphology, that is the outer structure of flower. Obviously, we'll be starting with that. We'll be starting with the structure of a flower. Let's keep it simple. Okay, we'll not be going to too much details, but of course, we'll discuss the outline of a flower's structure. And then later on, we'll in detail learn about the structure of anther, or in other words, the male reproductive organ, the female reproductive organ, and then we'll slowly move into fertilization. Okay, so before fertilization, we'll be discussing about the structures of the male reproductive organ and the female reproductive organ. Later on, we'll be discussing about pollination, okay, which is a mechanism that helps plants to reproduce. So we'll also discuss about pollination. After that, we'll be going into the topic of fertilization. And in the end of the chapter, we'll discuss about the post-fertilization events and about seed and fruit, and that's how we'll conclude the chapter. Okay, so I think you got an idea about how the chapter is going to be. Now let's start. Right, the first section that I'll be discussing here today is section 1.1, flower, a fascinating organ of angiosperms. You can see this in your textbook. Okay, if you have NCERT textbooks with you, you can see it in, this is the first section. You can see it in your textbook, right? So start writing notes with me. Okay, I'll show you a picture. Basically, that's how I'll be moving forward. I'll show you pictures, and that's how I'll be teaching. But pictures are not always the best way to learn, right? If you have a real flower in your hand, it would be much better, right? So if you have a flower around, okay, if you're living in your home, or if you're living in a place, if you're watching this video from a place where you can collect a flower, then go run outside and collect a flower. Okay, it can be any flower around you. Usually, I always learn with the help of the flower Hibiscus rosa sinensis. Right, I usually learn things with the help of hibiscus, because hibiscus is a typical example of a typical flower. Okay, so you can go take any flower that you want. I prefer hibiscus. Okay, so go run out and take a flower and try to find out the organs that I'm explaining to you. Okay, I'll be explaining, I'll be naming few organs over here, I'll be marking things. But meanwhile, you should also be able to look at the flower in your hand and identify which organ is which. Right? So let's play this game, right? Okay, moving forward. Before getting into the structure of the flower, let me tell you, what comes to your mind when we hear about the word, or when we typically hear flower? Okay, let me be precise. When you hear about, or when you see a rose flower, what comes to your mind first? In my mind, two things comes, right? First one is love, and the second one is affection, right? Usually love, affection, romance, right? Things like that are in most cases portrayed with the help of a flower like a rose. Okay, there are other flowers as well, but in general, red roses and stuff. Okay, cool. If you go to a marriage function, especially in India, if you go to marriage functions, you'll see a lot of roses, right? You'll have garlands with roses, you'll have bouquets, right? Roses and as well other flowers as well, right? You'll see a lot of flowers, okay? Even if you go to a mourning, right? If you go to a funeral, you'll also see flowers there, right? You'll see wreath and stuff, you'll see flower there as well. So flowers are basically involved in all aspects of our life. Okay, whether it is about emotions, whether it is about programs that we are having, from marriage to even death, we have flowers around us, right? We have flowers. Flowers. It's not about biology and reproduction, but flowers also has importance in our life. It has a social relevance, right? It symbolizes feelings, right? Human feelings like love, affection, happiness, etc., etc., right? So that's what it's like the first slide says. It's nothing important. Just getting along with the chapter, nothing more. Now let's move to the important part, the structure of a flower. Right. So here in this picture, it's figure 1.1 from your textbook itself. It's nothing new, nothing from like outside Google or anything. It's basically your textbook. Okay. So we'll be discussing some parts of the flower. Let's see. If you have a flower in your hand, and I suppose you do, just look at the flower and just look at the parts of it. Okay. You can obviously see some petals, right? If in the case of hibiscus, I usually take red hibiscus. I have red colored hibiscus at home. So the red colored part, or in usually most colored or like attractive part would be a petal in most cases. Okay, I'm not saying in all flowers it's like that. No, but in most cases the most attractive part would be petals, right? So you can see petals. Right, rose petals. We make things with rose petals, right? We collect rose petals, we preserve it and all that. Yeah, anyways, you can see petals, right? Petals are the first part that we'll be discussing, okay? Petals are actually of beauty. Petals are made for beauty, right? Petals actually, it helps in insect pollination. So petals actually helps in pollination. Note that point. Okay, why am I repeating this? Because we'll be discussing pollination later, we'll be discussing insect pollination later, and you should have an idea about how insect pollination works, right? So keep this in mind. Then the next part that we'll be having is sepal. The green part, usually it's not that prominent or big or anything. It's small and it's usually green in color. It's not that attractive or anything, right? You'll have sepals as well. In the case of hibiscus, you'll find 5 petals and almost 5 sepals as well, right? Okay, cool. Those are the two parts of a flower, petal and sepal, the first two parts to be exact. And these parts, the petal and the sepal, does not have any direct role in reproduction. So these are not reproductive parts of a plant. Okay, I said flower is a reproductive organ. Okay, it's fine. But inside the flower itself, the petals and the sepals does not have a direct role in reproduction. Of course, it helps in pollination and all, but it does not have a direct role in reproduction. So who has the direct role in reproduction? The male reproductive organ and the female reproductive organ. Right. The male reproductive organ is stamen. What is the male reproductive organ? Stamen. S-T-A-M-E-N. You have the word men in it, right? So just connect it like that. Stamen for man or men. It's usually the male reproductive organ. So the male reproductive organ is called stamen. And the stamen has 3 parts. Okay? 3 parts. So stamen is the male reproductive organ. Keep that fixed in your mind. The stamen further is divided into 3 parts, right? Those 3 parts are anther, filament, and connective. Connective is not marked in this figure, but you will see it later on. But anyways, there are 3 parts for a stamen: anther, filament, and connective. I hope that's clear. Okay, this is the male reproductive organ and it is directly involved in reproduction. Now moving on to the female reproductive organ. The female reproductive organ is called a pistil. Okay, what is it called? Pistil. P-I-S-T-I-L. Pistil. Or you have one more name for the female reproductive organ, which is Carpel. C-R-P-E-L. Pistil and carpel are often simultaneously used. They are like synonyms. Okay, so you should not confuse between them because we'll be learning about the parts of pistil later on. So you should not confuse between a part of a pistil and a carpel. Basically, pistil and carpel are both this exactly the same. Okay, it's used simultaneously. Fine. Pistil or carpel is the female reproductive organ, and this carpel or pistil also has three parts. Okay, the stamen also had three parts, right? Pistil or carpel also have three parts. Which are the three parts? The first part is the style. Okay, style. The second part is the stigma, and the third part is the ovary. You can see it in the figure, right? You can see stigma, style, ovary. That's the order that we usually learn. So learn it in that order itself: stigma, style, and ovary. Right? We'll go into more details later on when we when we have a detailed study about the male and female reproductive organs. But right now, just keep in mind that there is a male reproductive organ and it's called stamen. There's a female reproductive organ, it's called pistil or carpel, and you also have other accessory organs like petals and sepals. Okay, I hope that's clear. Now pause this video and look at the flower in your hand and try to identify which the stamen is, which the pistil is, or the carpel is, and the petals and the sepals. Count the number of petals and sepals for an activity. Okay, now moving on. Basically, that was our first section, 1.1, flower, a fascinating organ in angiosperms, right? It is indeed fascinating because without flowers, literally sexual reproduction wouldn't take place. It's only because flowers are present, sexual reproduction is taking place, right? So it is indeed fascinating. We can't argue on that. Anyways, moving on, we have the pre-fertilization events. So as I said, basically in this whole chapter, we'll be discussing about fertilization. We're studying about reproduction, right? What do you mean by fertilization? You've already heard this term before, I hope. Okay, you'll be hearing this in +1 maybe. Yeah, you have already heard about this term, but still I'll give you an idea. Fertilization basically means the fusing of male gamete and female gamete to form a zygote, and this zygote later develops into a whole new offspring. Okay, so that is what fertilization is. But we can't directly get into fertilization, can we? No, because fertilization is a very complex process and there are many steps to be taken before at the actual fertilization, right? Those steps which happen before fertilization is called pre-fertilization events. So here we'll be discussing about the pre-fertilization structures and events. What do you mean by pre-fertilization structures? Under the heading pre-fertilization, under the heading structures, you'll be learning about the male reproductive organ, stamen, and the female reproductive organ, pistil, in detail. From now on, I'll not be repeating male reproductive organ and female reproductive organ. Instead, I'll be using the term stamen and pistil. I hope you'll be, okay with it, right? Stamen is male reproductive organ and pistil is female reproductive organ. So let's start discussing the pre-fertilization structures and events. The first structure that you'll be dealing in pre-fertilization is the stamen. But before getting into that, let me ask you, you've all seen a flower bud, right? And you've all seen that flower bud growing into an actual flower. How does this actually take place? I mean, does the flower get to decide when this actually happens? Or does the flower actually undergo any change? Or does the plant undergo any changes before flowering? It does. It actually undergoes a lot of changes before an actual flower is seen. Okay, it undergoes a lot of hormonal changes. It undergoes a lot of developmental, structural changes. Actually, if a flower is actually present on a plant, you should understand that that plant has undergone a lot of hormonal and structural changes. So flowering is not a simple process. It is indeed complex. Okay, it involves the help of a lot of hormones, help of a lot of structures, right? So we'll be learning about all that in the later sections. But anyways, what you have to understand is that even before a flower is actually seen on the plant has already decided that it needs to flower. The process of flowering has actually begun much before the actual flower is seen on the plant. Okay, so that's what it's written over here. The plant decides to flower much before the actual flower is seen. Okay, the plant undergoes obviously many hormonal, developmental, and structural changes. What do I mean by developmental changes? Actually, you've seen a bud, right? But even before a bud, flower is formed from a bud. That's something everybody knows. But even before the formation of a bud, you'll be able to see a floral primordia. What is it? Floral primordium. Primordium typically is something that we see everywhere in the beginning stage. Okay, the term primordium usually depicts the beginning change of something, right? So here it is floral primordium, which means before flowering, right? Before flowering, before even the bud is formed, you'll be able to see the development of a floral primordium, right? And then this floral primordium later develops into a bud and then into a whole new flower. Okay, and these flowers can be either single or in the form of an inflorescence, right? What are inflorescence? Inflorescence are basically a collection of flowers. So if very small flowers are, in the form of a inflorescence, or if they are in groups, you literally call them inflorescence. Okay, so don't confuse the term inflorescence. Inflorescence is nothing but flowers, a group of flowers. Okay. Now, moving on, as I already told you, you have the androecium and the gynoecium, which we'll be discussing. What do you mean by androecium and gynoecium? A collection of stamens is called the androecium. So, stamen is just one single male reproductive organ. One single organ is called stamen. It's not necessary that one plant only has one stamen. No, some plants will be having more than one stamen. Okay, and some plants will be having more than one or two pistils. In the case of hibiscus, you can see a lot of stamens, right? They are even uncountable. You cannot actually count it. I mean, you can, it's possible, but it will take a lot of time, right? So anyways, in plants, the number of stamens and the number of pistil may vary greatly, right? And a group of stamen together, what do we call a group of stamen together? We call it androecium. And what do we call a group of pistils or carpels? We call it the gynoecium. Okay, I hope that's clear. Now let's move into the structure of androecium, or the structure of stamen, right? So that's our second subsection in the first section, which is 1.2.1, stamen, microsporangium, and pollen grain. So we'll be discussing about all these three: stamen, microsporangium, and pollen grain. So let's look at what a stamen is. You already know 2 points about stamen, right? The first point is that stamen has 3 parts. The first part is the filament, and the second part is the anther, and the third part is the connective. So a typical stamen usually has 3 parts. What do you mean by the anther? So anther is actually a lobular structure, which is usually in a globular shape or a kidney shape. The shapes can vary greatly, but it's usually a globular or a round or a spherical structure. It actually contains the pollen grain. Okay, it is from where the pollen grains are actually released. So you'll have the anther. Typically, you'll have 2 anthers. Typically, anther will be bilobed and all that. But anyways, it may vary greatly. Anyways, you have 2 anthers. In a typical stamen, you'll have 2 anthers, and those anthers will be connected to a long, slender stalk. Okay, you'll have 2 anthers or one anther which is bilobed, and then you'll also have a long, slender filament, right? And this long, slender stalk, it's known as filament. As I already said, it is known as the filament. And of course, the bulged part is the anther. Now, there should be a part connecting both the anther and the filament, right? That's known as the connective, as the name suggests, right? Connective is the part that connects the anther and the filament. The filament is the long, slender stalk of the anther or the stamen. Anther is usually where the pollen grains are stored and released. And there is a part called connective, which actually connects the filament to the anther. Cool. That's fine. That's one point that you already know about. The second point is something that you again know about, which is the number and size of stamen vary greatly. I've been mentioning this just a while before, right? The number of the stamen and even the size or the structure or the height or whatever varies greatly. It depends upon which species the plant is of. Okay. Now, about the anther. Let's talk about the anther, because filament and connective is not that important, because filament is just a slender stalk. It does not have any important role in reproduction, as same as a connective. But in the case of an anther is really important, because anther is where the pollen grains are produced, or the male gametes are produced, the male gametes are released. So we'll be discussing about all this, okay? How the male gametes are produced, how it is developed into a pollen grain, how it is released. We'll be discussing about all this. Before that, let's discuss the structure of an anther. How does an anther typically look? Just keep in mind that all these can differ. Okay, all these can differ. Don't come arguing that anther is not bilobed, anther is trilobed. Anther can be trilobed. Okay, it can be of multiple lobes. But the thing is, in a typical anther, it usually is bilobed. Okay, what do I mean by bilobed? What does it mean by bi? Bi means 2. Bi, di, all these terms literally mean 2, right? So usually one anther has 2 lobes. Okay. I usually study an anther in the form of a house. Okay, I imagine that the anther is a house. with 2 rooms. Okay, I imagine an anther as a house with 2 rooms. Okay, so one house has 2 rooms, and there is another house nearby which also has 2 rooms. That is how an anther is. Okay, so how is an anther? Anther is like 2 houses with 2 rooms each. So you have one house here and you have another house here, and both these houses has 2 rooms. Okay, this is how a typical anther is. So we'll convert this into biological terms. So literally these 2 houses are the 2 lobes of an anther. So I already told you that an anther is bilobed, right? So these 2 houses can be imagined as 2 lobes. Okay, I'm not sure if you can see me drawing the house and everything. Okay, I'll read or draw it over here. Okay, you have 2 houses, right? You have 2 houses, and both these houses has 2 rooms each. Okay, it has 2 rooms each. So imagine these houses as the lobes of an anther. So how many houses do you see? 2, right? So there are two lobes, 1 and 2, and in each lobe you have 2 theca or 2 room. Okay, theca is room is depicted as theca. Okay, anyways, you have 2 rooms inside each lobe. So you have 2 lobes in an anther, and each lobe will be containing 2 theca. It's that easy to study. Just imagine 2 houses with 2 rooms each. Okay, so that is what is written over here. You have an anther which is bilobed, and each lobe has 2 theca, right? So it's dithecous. Bi and di usually means 2. So an anther is bilobed, and each lobe will be having 2 theca, which means it's dithecous. Okay, so we can imagine anther as a tetragonal structure or as a 4-sided structure. Like if we put a drone about both these houses and we look into it, we can see 4 rooms, right? 4 rooms located at 4 sides. So basically an anther is a tetragonal structure or a structure which is 4-sided. Okay, I hope you agree with that. Now, at these 4 rooms, you have the microsporangia. Microsporangia and theca are all simultaneously used. It doesn't differ much, so you may not confuse with all that. Just understand there are 4 rooms, right? These 4 rooms can be called theca. Also, it can also be called microsporangia or microsporangium in singular. Microsporangia is the plural term. You have 4 microsporangia at 2 corners. Right, 2 in each lobe, as I said. It's similar as theca. The arrangement is same as theca. A theca later develops into a microsporangium, or it is usually simultaneously used. Anyways, the term that you have to remember, the point that you have to remember is there are 4 microsporangia inside a bilobed anther. Okay, in each lobe you will have 2 microsporangia. I hope that's clear. So that is how the basic structure of an anther goes. Now moving on, these microsporangia later develops into pollen sacs. What is pollen sacs? Pollen sacs are basically where pollen grains are stored and formed. Okay, later they'll be released from those pollen grains, pollen sacs. So basically, these pollen sacs are somewhere where the pollen grain is stored or released from. Okay, these pollen sacs are developed from these microsporangia. Okay, so how many pollen sacs will be there? I already told you that there are 4 microsporangias, right? There are 4 microsporangias in the bilobed anther. And I also told you that the microsporangia is from where the pollen sacs develop. So how many pollen sacs will be there? 4. I'm not talking about pollen grains here. I'm talking about pollen sacs. Pollen grains, if you count the number of pollens, you can't count the number of pollen grains, right? There will be numerous pollen grains from a single anther or in a single pollen sac. But how many pollen sacs will be there in a typical anther? The answer is 4. Okay, you'll have 4 microsporangias which will later develop into 4 pollen sacs. That's like common sense, right? This is usually the normal structure of a stamen. I'll show you an image. So this is the image that you have in your NCERT textbook. So you can see the lobed structure, anther over here. You have the long, slender filament or the stalk, right? And if you zoom into the anther, you can see a tetragonal structure. Here you have the first theca, the second theca, right, the third theca and the fourth theca, and these 2 are in one lobe and these 2 are in another lobe, right? So you have 2 lobes and 4 thecas or 4 microsporangiums, right? Inside those 4 microsporangiums or pollen sacs, you can see pollen grains as well. I told you microsporangium later develop into pollen sac, right? So in those 4 pollen sacs, you can see pollen grains as well, right? So this is how an anther looks. Okay, typical anther looks. Now moving on to the next structure, which is a microsporangium. How does a microsporangium look? So microsporangium are basically the rooms inside those houses which we've been talking about, right? So they are basically those rooms which has the pollen sacs, or which has the which are the pollen sacs which contain the pollen grains. Cool. But these microsporangiums are actually covered with a lot of wall layers. Just imagine inside your house, if you had a treasure, if you had something very precious and if you wanted to save it, you would obviously protect the house really well, right? So inside a microsporangium, you're seeing the pollen grain. And inside the pollen grain, you'll be having the male gamete. Okay, so that is something really special. Right, it is very precious. Without the pollen grain, reproduction will not happen. Right, so pollen grain is really special and precious as far as reproduction is concerned. So this is something that should be clearly protected. Right, so this microsporangium actually has 4 wall layers, and this is one of the most important topics in your chapter. Okay, because I've seen many questions, many previous year questions from this single headline or this single topic. Okay, microsporangium or its walls. You'll also be asked about anther walls. It's typically the same thing. Okay, anther walls. If you're asked about anther wall, you can write the same thing. It's typically the same. Okay, it's basically the same. Now moving on, you have 4 wall layers. As I already told you, you have four wall layers. Which are they? The first one, the one at the periphery, is the epidermis. Okay, so I'm not that good at drawing, but I can try. Okay, so you have the outer part, which is the epidermis, and then you have the inner wall layer, which is the endothecium. And as we go inside, you have middle layers, which can be 1, 2, or 3, depending upon the species. And at the inner side, at the innermost side, you will have the tapetum. So you will have the epidermis at the outer side, that is the outermost wall layer, the epidermis, and then you'll have the endothecium, and then you can see some middle layers. Middle layers can be 1, 2, or 3, it depends upon the species. As I said, you have the middle layers, and then later on, and the innermost part, you'll have the tapetum. Tapetum. Okay, know the point because it's really important. So you have the anther or the microsporangia has 4 wall layers. Which are they? Come on, say it with me. Epidermis, endothecium, middle layers, and tapetum. So middle layers can be 1, 2, or 3. So don't ask me whether it will be like 5 or 6 wall layers then. It will be. But as we name them, there are only 4, right? The first one is epidermis. The second one is endothecium. The third one are middle layers. And the fourth one is tapetum. Right. The most important one in all of these is tapetum. Why is it so important? We'll see. Something is written over here. Let's see what it is. Tapetum has a dense cytoplasm. Okay. So tapetum are basically, it's a wall layer. It's basically made up of cells, right? Whatever it's there inside a plant or a human body, it's basically made up of cells. Okay, so you'll have cells inside this tapetum as well. And those cells will be having a dense cytoplasm. The cytoplasm will be so thick and so dense. You'll also have more than one nuclei. So it's not like the tapetum cells are not like this, but the tapetum cells are like this. You have more than one nuclei and the cytoplasm is kind of really dense. Okay. Now moving on to the function of these 4 wall layers. Before that, I'll show you a picture. Again, another NCERT picture, figure 1.3. You have the A part, which actually shows how the connective lies, how the epidermis lies, the endothecium lies. It's just an outline. Okay, you can see the epidermis here, which is the outermost layer. Then comes the endothecium. Then you have the middle layers, and then the tapetum. And inside, something else is marked. What is it? Sporogenous tissue. What is sporogenous tissue? We'll discuss later on. Fine. Okay, we'll discuss later on. Just wait for a sec. Now, just you'll have to concentrate now on the wall layers. Which are the wall layers? Is sporogenous tissue a wall layer? No. There are only 4 wall layers, and they are epidermis, endothecium, middle layer, and tapetum. Let's see a zoomed image. Right. The first one, as I already said, you have the epidermis, you have the endothecium, middle layers, you have the tapetum, and just look at the tapetum cells in the figure. Okay, the tapetum cells, it's clear that you have multiple nuclei over here, right? You have multiple nuclei over here, and the cells, the cytoplasm is really dense. Okay, cool. I hope that's clear. Now moving on, let's talk about the functions of these 4 layers. These 4 wall layers are not just there for, they really have a purpose, okay? They're not like just for a beauty or just for protection, but they really do have factor. They really do have a purpose, right? So the first 3 wall layers, okay, from the outside, from the periphery, the first 3 wall layers has the function of protection. Okay, it's basically common sense, right? So if we are like building a fort, why would we build a fort? Of course, for protection, for defense, right? So the first three, from the periphery, the first three wall layers, so the outer three wall layers will be for protection. Okay, the first function is protection. And what is the second function? The second function that is that it helps in the dehiscence of anther. What do you mean by dehiscence of anther? Dehiscence of anther simply means the anther has to break someday, right? The anther has to break and the pollen grains has to come out. It's only then reproduction will happen. Will reproduction happen if the pollen grains lies inside the anther? No, the anther obviously has to break one day and the pollen grains has to come out. So these three wall layers, the wall layers from the periphery, help in the dehiscence of anther or in the breaking of anther. Okay, but the tapetum has a completely different function. The tapetum is the innermost layer, so it will not be having any function in protection, which is again common sense. So the tapetum has a function of nourishment. Okay, tapetum provides food or provides nourishment to the developing pollen grain. So inside the anther or inside the microsporangia, a pollen grain is developing, right? A pollen grain or a male gamete is developing, or male gametophyte is developing, right? And this male gamete obviously needs food. Okay, the male gametes, there'll be two male gametes, and these male gametes obviously needs food, right? So the nourishment is given by this tapetum. So now you can connect. Now you can connect the point from before, the slide before, right? The tapetum has a dense cytoplasm and multiple nuclei. Of course, it's a wall layer that provides nourishment. It should have dense cytoplasm and multiple nuclei, right? So tapetum, the function of tapetum is really very, very, very important. Tapetum is the innermost cell and also it provides nourishment to the developing pollen grain. I hope that's clear. So now you have the epidermis. You have the endothecium, you have the middle layers, it can be 2 or 3 or 4, and then you have the tapetum, and inside all this you will be able to see some cells. Okay, you will be able to see some cells. These cells are known as the sporogenous tissue. A group of cells is known as a tissue, right? So you can see many cells, and these cells will be named the sporogenous tissue. Sporogenous tissue. Wait guys, did I make a mistake before? No, right? Yeah, that's tapetum itself. Anyways, inside the tapetum, you'll have the sporogenous tissue. So what is the function of a sporogenous tissue? I'll tell you. I told you that inside the microsporangium there will be a pollen grain. Pollen grain doesn't come from the earth, from the sky, right? It doesn't like come out of nowhere. It has a development process. A pollen grain or a male gamete develops from these sporogenous tissues. The cells inside this sporogenous tissue, or the cells inside this sporogenous tissue develops into a new pollen grain or a male gamete. So let's see how how that development happens. Okay, so what is a sporogenous tissue? It's a group of a tightly packed homogeneous cells. What do you mean by homogeneous? Cells that look alike. Okay, cells that have a similar structure, a similar size. See the figure I've drawn here. All the cells actually have a similar structure, okay, and it also will be almost of the same size, not exactly but almost of the same size. Okay, so sporogenous tissue is basically a group of cells which are homogeneous and which are tightly packed. You'll not have any intercellular spaces in between. Cool. So that is the 4 wall layers, and inside you'll also have the sporogenous tissue. Now let's look at how the sporogenous tissue develops into a pollen grain, or simply saying into a male gamete or a microspore. Okay, all these terms are kind of like synonyms. Okay, they have slight differences. I'll tell you what it is. But simply saying, this sporogenous tissue or the cells inside the sporogenous tissue develop into a microspore. So what do you call the process of formation of a microspore? What do you in general call the a process of formation? You call it genesis, right? So something that is termed as genesis literally means formation. So what is formed over here? Microspores. So it's known as microsporogenesis. So what is microsporogenesis? It's simply the process of formation of microspores from the cells of a sporogenous tissue. It's defined over there, right? The process of formation of microspores from a pollen mother cell. What do you mean by a pollen mother cell? The cells that undergo meiosis, or the cells that develop into a pollen grain or a microspore, is called the pollen mother cell. Let me make it clear. I said there are sporogenous tissue with many homogeneous cells, right? And these cells participate in the formation of microspores. So what happens is, I'll just represent one cell over here. So there are a lot of cells, okay? Out of this, one cell, one or two or three, you almost all cells develop into microspores, okay? So you'll have, I'll only be showing you the image of one cell or the diagram of one cell, but you should understand the same thing happens with all other cells, okay? So one cell is now ready for undergoing microsporogenesis. So what is the first step of microsporogenesis? It is meiosis, okay? What is the step? Meiosis. So this cell, which is ready to undergo microsporogenesis, is called the pollen mother cell, or the PMC. So what is a pollen mother cell? Pollen mother cell is nothing but a cell of the sporogenous tissue which is ready to undergo meiosis, or which is ready to participate in microsporogenesis to form a microspore. So that's what a pollen mother cell is. So this pollen mother cell, okay, I'll draw a more bigger figure, okay? I'll just show you how a pollen mother cell turns into a microspore. So this is a pollen mother cell over here, PMC. And what will be the ploidy of this pollen mother cell? You already know what ploidy is. You have learned a lot about haploids and diploids, right? So what will be the ploidy of a pollen mother cell? Usually it will be diploid in nature, right? In the case of diploid plants, the pollen mother cells will also be diploid. Okay, these pollen mother cells undergo meiosis. What does it undergo? Meiosis. So meiosis, as you all know, is reduction division, right? It is reduction division. So what happens? The ploidy gets reduced into the half, right? So if it's diploid, it reduces into direct half, which is haploid, okay? So after meiosis, you will get 4 microspores, which are all haploid in nature. So what will you get as a result of microsporogenesis? As a result of microsporogenesis, you will get 4 microspores, which are all haploid in nature. Where does it come from? It comes from a single pollen mother cell, which was diploid. And what is the process in between? Meiosis. In the beginning, in the initial stage, after the meiosis, these 4 microspores are present in the form of a group, or these 4 microspores stay together, they stay close to each other. That's what is called a microspore tetrad, okay? So what is a microspore tetrad? Microspore tetrad is nothing but the 4 haploid microspores staying close to each other, okay? Later on, they separate and form 4 separate haploid microspores, okay? So don't confuse between these terms. It's not anything huge or anything complicated. Simply saying you have this sporogenous tissue, and this sporogenous tissue has many cells which are homogeneous in nature or which look alike, and each of these cells are called the pollen mother cells, and they are ready to undergo microsporogenesis or meiosis. So how does it take place? A pollen mother cell, which is diploid in nature, undergoes meiosis. And what does it give you? It gives you a microspore tetrad in the beginning. In the initial stage, it gives you a microspore tetrad, right? And this microspore tetrad then gets separated into 4 haploid microspores, okay? And the process in between is called the meiosis or the reduction division. So I hope that's clear. So this is what microsporogenesis is, okay? It's nothing complicated. Simply, the thing that you should know is that there will be a pollen mother cell which will be diploid, and from that pollen mother cell, after the process of meiosis, you will get 4 microspores which are all healthy and good to go. These microspores later develops into a pollen grain. Okay, the microspores will not be directly released into the environment, but it will be converted into, or it will develop into a pollen grain, and only the pollen grain will be released into the environment. Okay, so what is a pollen grain? How does a pollen grain look? That's what we'll see. A pollen grain is simply a male gametophyte. What does the term gametophyte mean? Something that carries a gamete is called the gametophyte. So if there is a pollen grain, it will be having the male gamete inside. Usually one pollen grain will be having 2 gametes inside. Okay, so imagine a pollen grain which 2 male gametes inside. Okay, there will be a pollen grain and 2 male gametes will be inside that pollen grain. Okay, so the pollen grain is just a body which carries the male gamete, right? So something that carries a gamete is called a gametophyte, and of course it is carrying the male gamete, right? So it's known as the male gametophyte. What is it called? Male gametophyte. Okay, so the pollen grain is technically not the male gamete. Pollen grain and the male gamete are not equal. Okay, it's not the same. Pollen grain is actually just a body which carries both these male gametes. Okay, so if you have a pollen grain, there will be 2 male gametes inside, which is why the pollen grain is called the male gametophyte. The pollen grain is not the male gamete, but instead it is the male gametophyte. It's just a body which carries the male gamete, right? Pollen grain will usually be spherical, as I have drawn right here. The pollen grain will be usually spherical in shape, and the diameter would be 25 to 50 micrometers. I'm not talking about meters, I'm not talking about centimeters, I'm not talking about millimeter, I'm talking about micrometers here. It's that small. Okay, a pollen grain will typically have a diameter of 25 to 50 micrometer, and it will be spherical in shape. Now we'll be discussing about the wall layers of a pollen grain. So pollen grain is again a very important part. It carries 2 male gametes. So of course the male gametes should be well protected. So it will have a wall layer outside. And these wall layers actually contain, these wall layers are composed of 2 different types of layers. Okay, let's see what it is. Imagine this is a pollen grain. Okay, you'll have one layer like this. Okay, which is kind of smooth and without any, any disturbances. It will be smooth and kind of thin and all that. And then you have another layer in this shape. Okay, the inner layer is called the intine. The inner layer is called the intine, as the name suggests. Inner layer, intine, okay, the inner layer is known as the intine, and the external layer or the outer layer is called the exine. Calling it avine would be a bit weird, so that's why we call it exine. Okay, because the outer layer can't be called avine, that would be weird, so we're calling it the exine because it's the external layer. So you have the inner layer, which is the intine, and the outer layer or the external layer, which is the exine. This exine is made up of a very strong compound which is known as the sporopollenin. It's a very strong chemical compound known as the sporopollenin found in nature, which is actually so resistant to any kind of enzymes or temperature fluctuations, rain, water, whatever. Okay, it's really resistant. The sporopollenin is really resistant to action of any enzymes or temperature rise or humidity or moisture or whatever. The sporopollenin doesn't care for it. Okay, the sporopollenin is actually very resistant and it is not easily destroyed. And that's exactly why you have it in the exine of a pollen grain. Okay, so what is the exine of a pollen grain made up of? It is made up of sporopollenin. Okay, it's a chemical compound. And the exine has germ pores. What is germ pores? So germ pores are some parts, okay, I'll just zoom in here. If it's like this, okay, if the wall layers will be like this. If I'm zooming in, the exine will be like this, which means there will be a small part which is not covered with sporopollenin, and those parts are called the germ pores. Okay, so you have the exine which is made up of sporopollenin, but there will be small parts or holes or pores which are not covered with sporopollenin, and that's called the germ pores. Okay, that's all about the exine. Now about the intine. Intine doesn't have any germ pores. It is continuous, it is smooth, okay, and it is made up of cellulose and pectin. Okay, it's not made up of sporopollenin. Of course, the exine is made up of sporopollenin, but the inner layer or the intine is made up of cellulose and pectin. It's comparatively thin. In this figure, I've drawn it a bit thicker. Don't mind that. Okay, the inner layer is usually thinner than the exine. Okay, the exine is more thicker and the intine is more thinner. Okay, don't confuse between them. The intine is more thinner because it's the inner layer, so it'll be thinner, but the exine is the outer layer, so it'll be more far stronger and thicker, right? That's common sense again. So the intine is made up of cellulose and pectin, and the outer layer, which is the exine, is made up of sporopollenin. Okay, and inside this intine, you'll have the plasma membrane, or simply saying you'll have the male gametes, right? You'll have the male gametes, and those male gametes are basically cells, and cells, of course, have the plasma membrane, right? Okay, so this is what the structure of a pollen grain looks like. I'll show you an image. I'll show you the image of real pollen grains over here. They are electron micrographs of few pollen grains. It's obviously from your NCERT textbook. See the structure. It kind of looks like coronavirus, right? We used to draw corona, or represent coronavirus like this in figures and all that. But anyways, it has an intine which is made up of sporopollenin, which has germ pores as well, and sorry, an exine with made up of sporopollenin, which has germ pores, and an intine which is smooth, and the inner layer which is made up of cellulose and pectin. So I hope you are clear with the structure of a pollen grain.